So basically I stand here and use the mouse to point out.
Yeah, that's it.
But if I just, I see. Okay, so can I do this?
Howdy. I'm Chen, first-year PhD student from Guo's lab. It's my pleasure to introduce today's speaker, Dr. Boyi Gan. Dr. Gan received his bachelor's and master's degree from Fudan University in China and completed his PhD study at Cornell University. He later pursued advanced training at Dana-Farber Cancer Institute. Currently, he is a tenure-track assistant professor in Department of Experimental Radiation Oncology at University of Texas MD Anderson Cancer Center. Dr. Gan mainly focuses his research on understanding the biological process linking energy sensing metabolism to cancer and stem cells. Current work in Dr. Gan's lab is aiming to study PI3K, AKT FOXO, and LKB1 AMPK pathways and the roles of long non-coding RNAs in energy sensing, cancer metabolism, and tumor suppression. Today, Dr. Gan will give us a lecture titled Energy Sensing and Metabolism in Cancer and Stem Cells. Please join me in welcoming Dr. Gan.
So it's a great pleasure to visit Texas A&M, and I want to thank Shaodong for the invitation and also the student for the nice introductions. So what I would like to do first is to give some broad pictures on our research background. So as we know, one key feature to distinguish us as a living organism from those other matters such as stone is the capability of the living organisms to be able to sense and respond to the alterations in their surroundings, including the fluctuation in the metabolic, the metabolites and energy levels in our surroundings. So I have been, keep a long-standing, we have a long-standing interest in the questions such as how cells sense and respond to the energy and the nutrient availability. So these are very fundamental questions. But also in the context of cancer biology, how cancer cells adapt to survive and grow under metabolic stress. Now, when we talk about energy metabolism, probably one of the most important molecule is ADP. So this is so-called the molecular unit of currency of intracellular energy transfers. And we all know that the ADP hydrolysis can release the energies to support essentially every cellular processes, such as protein synthesis or muscle contraction, just to name a few here. Now, it is absolutely critical for cells to be able to balance the energy intake and energy expenditure at both organism level and cellular levels. So we know that dysregulation of such balance leads to many diseases. I think the most probably obvious link is obesity and diabetes, right? So, but we are particularly interested in its connection to other diseases such as cancer and also some of the diseases associated with stem cell defects. So indeed, our cells have designed a very elegant system to help us to sense the energy levels, to help us to adapt to, what if there is not enough energy, what we should do, right? So this is mediated by a very critical energy sensor we call energy sensor, the AMP activity protein kinase or AMPK. So what happens here is that if there is energy stress, say, if you catch a cell under the glucose-free medium, or as what I'm experiencing right now, if you give a seminar during the noon time before you have the lunch, so that is going to induce energy stress. Specifically, that's going to deplete the ATP levels, but also increase the corresponding the AMP level in the cells. Now, it is the AMP which is essentially sensed by AMP activity protein kinase, as you probably can tell from its name. So what happens here is the AMP can interact with AMP kinase, and that leads to its allosteric activation. Now, the AMP kinase is, as its name tells you, is a serine kinase, and so its full activation requires its phosphorylation by upstream kinase called LKB1, and I will come back to this later. So this phosphorylation leads to its full activation. So once it is activated, it will then phosphorylate a bunch of downstream targets, and this basically serves to two purposes. First is to promote those ATP generating catabolic processes, but meanwhile to shut down those ATP consuming anabolic processes. So what happens here is basically cells trying to restore the energy balance if there is not enough energy, so to support the cell homeostasis. So we know that one of the probably the process, anabolic process, which consumes a lot of energy is protein synthesis. So this is mainly mediated by another key mediator called mTOR complex 1, or mammalian target for rapamycin complex 1. So what happens here is if there's energy stress, the NPK is activated, the NPK can phosphorylate some of the components involving mTOR pathway, such as TSC2 and Raptor, which is not shown here, but is a component of mTOR complex 1. And this eventually serves to inhibit mTOR 1 complex function and to suppress the protein synthesis and cell growth and also cell size increase. So this actually makes sense, right? Because you think about, because protein synthesis consumes a lot of energy, so if there's not enough energy to support such expansion, then the cells, they make decisions. So they tell the cells, we don't have enough energy, so you'd better be very careful, don't make too many proteins, because that's going to consume a lot of energy, and we cannot afford it. So this is a very fundamental mechanism here, but actually is of high relevance to human diseases. I mean, diabetes, the obvious, there's many studies to study NPK pathway in the diabetes, the metabolism syndromes, but also cancer. And the first line of evidence came from the initial analysis shows that the LKB1, which is the upstream kinase I just mentioned of AMPK, is mutated in several human cancers, that including a hematoma cancer syndrome called the Peutz-Jeghers syndrome, and also several others sporadic cancers, such as most notably non-small cell lung cancers. Now, its downstream targets, such as TSC, is another key tumor suppressor genes, which is mutated in another hematoma cancer syndrome called tuberous sclerosis, and also mutated in several other sporadic cancer types, such as kidney cancer and bladder cancers. So both upstream kinase and the downstream effect of MPK are mutating in human cancers are bona fide tumor suppressors. So that providing a strong evidence to link this pathway to human cancers. So we are interested in studying the regulatory mechanism of this energy sensing signaling pathway in tumor suppression and also stem cell homeostasis. So initially, why we're interested in this pathway, function of this pathway in stem cell biology? Because at the time when we initiated this project, we already know that, for example, cell cycle is critical to maintain the stem cell homeostasis. Some of the developmental pathways, such as Wind pathway, are well known to regulate stem cell homeostasis. But it remains largely unknown whether the energy metabolism or energy sensing process play any role in stem cell homeostasis. So we set up to answer this question by using the mouse genetic approaches. So first I want to give only a very brief introduction on what is stem cells. So basically, particularly here is the relevance. We use the hematopoietic stem cell as a model system to study our questions. So hematopoietic stem cell basically the type of adult stem cells which function to replenish the whole blood lineages in our cell, throughout our lives, right? And it's mainly reside in the bone marrow systems. So this whole process of blood cell replenishment is also called hematopoiesis. So what happens here is the hematopoietic stem cell, they need to self-renew themselves, meaning duplicating themselves without losing developmental capability. Meanwhile, the hematopoietic stem cell also can differentiate into various lineages, as shown here, such as T cell, B cell, etc. And those cells function as effectors to serve to in our bone marrows and peripheral blood systems to function to sustain the hematopoietic functions. Now, one key feature of hematopoietic stem cell is the stem cell needs to reside in a relatively quiescent stage, meaning the cells, they don't proliferate very much. So normally they remain in a less proliferative stage, and that's critical for its maintaining its self-renew capability. But nevertheless, the stem cells can be activated to quickly re-enter into the cell cycle to promote hematopoiesis. Now, so the question at the time is the balance, how the hematopoietic stem cell balance between the self-renew versus differentiation quiescence versus proliferation is always a critical question in the field, and we ask the question whether the energy sensing checkpoint play any role in this process. So again, we address this question using the mouse genetic approaches. So since this has been published a while ago, so I'm just going to very briefly summarize our finding. So we found that both the upstream kinase of LKB1, upstream kinase of MPK, LKB1, and also downstream effector of MPK, TSE, play critical role in maintenance or in the maintaining hematopoietic stem cell homeostasis. So because this is published a while ago, here I'll just show some of the key data here. In this study, we used the LKB1 conditional knockout mouse models in which we can induce the LKB1 deletion in the adult mice by simply treating the mice with tamoxifen, so that can induce the activation of CREAT2, which can then mediate the targeted gene deletion. So in this case, we induced the LKB1 deletion in the adult mice, and we found a very striking phenotype in the adult mice. That is, we see a very dramatic reduction of the hematopoietic stem cell pools. So the hematopoietic stem cell quickly depleted after we delete LKB1 in the adult mice. So shown here actually is representative data which shows the so-called the SLC cells, which is representing the c-KIT and SCAR1 double positive and lineage negative population, which is kind of a mixed population of hematopoietic stem cell. And you can appreciate there is a sharp decline of the hematopoietic stem cell pools after we delete LKB1 in the adult mice. And this phenotype is associated with acute anemia, pancytopenia, meaning the loss of all types of blood cells in the hematopoietic organs. And eventually the animal dies within several weeks after we deleted LKB1 in the adult mice. So another golden standard assay to test hematopoietic stem cell in vivo is to conduct the bone marrow transplantation assay. So in this case, we did a 1-to-1 competitive transplantation in which we mix the LKB1 wild type or knockout donor bone marrow cells with competitor cells, which is the wild type cells. So you look at basically, so you transplant those cells into the recipient mice, and then look at how they compete with each other using the FACS analysis. So, and you can appreciate that initially you can see there's roughly about 50% reconstitution after we transplant either wild type or knockout cells into the lethally irradiated the recipient mice. But then you can see there is a very dramatic decrease from about 50% reconstitution down to less than 5% reconstitution only after 4 weeks of the transplantation. So this clearly shows that the LKB1 deletion has a very dramatic effect on the reconstitution of those hematopoietic stem cells in the bone marrow systems. So suggesting that LKB1 deletion results in a reduced hematopoietic stem cell reserves. So I'm going to skip a lot of data because again, this is published a while ago, but the take-home message from our study is that LKB1 mediated energy sensing is critical for hematopoietic stem cell maintenance, which also points to important coupling or integration between the energy availability and the stem cell homeostasis. So mechanistically, we found that LKB1 can regulate the energy metabolism in the hematopoietic stem cells through different mechanisms. One of the downstream effect obviously is AMPK, but others including, for example, is regulation of PGC1, which leading to the regulation of mitochondrial biogenesis. So now I'm going to, so one last point is that, so this suggesting that, if you can improve, so this suggesting that the AMPK mediated energy sensing checkpoint is important for us to maintain the stem cell homeostasis. And we know that stem cell homeostasis also plays a very important role in longevity, in aging process. So indeed, there has recently actually there is on the market there is MPK activator. So if you go to let's say Walmart, actually you can buy this from the supermarket. So basically, this has been proposed to this activator has been proposed to promote use for metabolic health. So that at least a part of the mechanism probably is because it can promote the stem cell functions in humans. So that's just to show some of our implications, the study of our implications, implication of our studies in human disease. So I'm going to, in the rest of my talk, I'm going to focus on more recent study on energy synthesis and metabolism in cancer. Again, the question we are asking is how normal cell or cancer cell sense the energy availability, how cancer cell adapt to survive and grow under energy stress condition, and eventually we want to translate our understanding of energy metabolism in cancer into novel cancer therapeutics. So first, let me just give a little bit background introduction on the connection of energy metabolism to cancer development. Actually, one of the most prominent connection came from epidemiologic study showing that obesity is the second greatest risk factor for cancer development in this country. So we know the number one risk factor is smoking, right? So, but probably less appreciated is obesity actually is the second greatest risk factors. And it has been estimated in this country each year about 80,000 cancer-associated deaths can be prevented if all of us can maintain our BMI within 25. So if we can have a healthful living style, actually that can significantly decrease the cancer incidence. But then also in the last few years, it has been more and more appreciated that the deregulated energy metabolism play a very fundamental role in the cancer biology. Indeed, this has been included as a new hallmark of cancer. And finally, some of our understanding on the energy metabolism in cancer has been started to be translated into cancer detection and treatment. So I'm going to just mention two examples here. So first is the FDG PET. So this is a frequently used clinical practice to image the tumor, incidence and also its metastasis to other organs by looking at the glucose uptake in the cells. So this is because the Warburg effect, probably some of you know that. So the tumor cells, they tend to uptake a lot of glucose. So here we use the fluorescence labeled deoxyglucose, which is glucose analog. So it can be uptaken into the cells, but it cannot be further processed to downstream glycolysis pathways. So in this case, they stay in the tumor cells. So the higher fluorescence labeled the deoxyglucose indicate the more likely there is a tumor cell there. So in the clinical setting, often use FDG PET to image the tumor, to detect the tumors in the patients. Now, in terms of treatment, a well-known example, I think, for people study diabetes or type 2 diabetes, probably know this better than me. That is this drug called metformin, because this is a widely used drug to treat diabetes. But in our, in the cancer field, now in the recent years, there has been a lot of studies to study the role of metformin or fenformin in cancer prevention or cancer the tumor biology, largely because there is evidence to show that the metformin treatment is associated with a decreased tumor incidence and mortality. So I'm going to come back to this point later during my talk. So again, as I mentioned earlier in the beginning, that obviously NPK is one key player involving energy stress signaling pathway. It functions as an energy sensor to sense the lack of energy. It helps the cells to restore the energy balance in response to energy stress. I also mentioned that NPK function to repress tumor development, at least in some settings in human cancers. So we are continuing to study the recurrent mechanism of NPK function in response to energy stress and how it's relevant to cancer biology. I'm going to introduce one recent study from our lab to link non-coding RNA to NPK-mediated energy sensing checkpoint. So first I'm going to just give one slide introduction on what is non-coding RNA. I think probably most of you probably have heard of this, but this is basically a summary from our recent publication to discuss the role of non-coding RNA in cancer metabolism. But suffice to say, the non-coding RNA or lincRNA is one type of non-coding RNAs which are long. So by definition, there are more than 200 nucleotides. It has very different function from another type of non-coding RNA, such as microRNA, which are usually short. So they have also mechanistic, they are very different. So non-coding has very diverse functions in different biologies. So it can function to interact with transcription factor or epigenetic regulator in the nucleus, thus influence transcriptions. It can also function in the cytoplasm to function as, for example, molecular sponge to sequester a protein or RNA from binding to other biomolecules, so that can regulate its downstream signaling pathways. In general, its function in cancer metabolism, particularly in the energy metabolism, energy synthesis process, still remains largely unknown. So we have taken effort to understand the non-coding RNA function in cancer metabolism or in energy stress response. In this case, we have taken an unbiased approach. We try to identify those non-coding RNAs which can be induced upon metabolic stress conditions, such as under glucose starvation condition, or can be induced by certain transcription factors which have been known to play an important role in metabolic stress response, such as FOXO, as I'm going to mention later. Then we conduct the RNA sequencing coupled with sophisticated computation analysis to identify those unknown non-coding RNAs which can be induced under those conditions. And we then filter, so you can identify many such non-coding RNAs from such analysis. But to link this to cancer biology, so we filter the list of those non-coding RNAs to TCG analysis. So this is the cancer genome atlas analysis. So you see all those genes go up and down, how they altered in the cancers. So we will have a sense whether this non-coding RNA is, somehow altered in human cancers. So we try to provide some rationale, to study those non-coding RNA in cancer. So then we generated a list of non-coding RNAs which, we know they are altered in human cancers. They always are induced by metabolic stress condition or transcription factors. Then we subject them to a series of functional analysis platforms in our lab, such as we functionally validated their function using in vitro and in vivo studies. We study their metabolism functions. We also study their molecular mechanism. So, for example, how it regulates different proteins or its downstream targets. And also, finally, by collaborating with the clinician at MD Anderson, we also study its clinical relevance. So we will apply this, all these different approaches in the following studies I'm going to mention. So one of the non-coding we identified from this study is a non-coding which can be induced upon glucose starvation. So glucose, as I mentioned earlier, is one major metabolite or nutrient which can be utilized to provide the major carbon source. So if you deprive cells from glucose, that quickly can deplete ATP, can induce energy stress. So that's commonly used in our cell culture study to induce energy stress. Of course, we know glucose also play a very important role in insulin resistance, for example, in the in vivo setting. So very, very important molecule here. Now, what we have done here is, again, we culture cell in glucose-containing glucose-free media. We did RNA sequencing, did the computation analysis, and here we identified a bunch of non-coding RNAs. But here we're particularly interested in this non-coding RNA called NBR2, for neighbor of BRCA1 gene number 2. So why we are interested in this particular non-coding RNA? Because this non-coding RNA looks interesting from its locus. So shown here is a BRCA1 gene. As you can tell from the name, it has something to do with this gene called a BRCA1. So if you know cancer biology, probably you already know this gene. This is probably one of the most famous gene to the public, in terms of one of the most famous cancer gene to the public, because it's associated with the breast cancer, and so it's very, very famous tumor suppressor genes. Interestingly, in the mouse genome, there is another gene called NBR1, which resides right next to BRCA1 gene. So there is only a couple of hundred base pairs between these two, the transcription start sites of these two genes. Now, then during the evolution, there is a genomic duplication, the subsequent alteration in this locus. So end up in the human genome, the BRCA1 gene now is right next to another gene called the NBR2. So this NBR2 is a partial duplication of NBR1. So the 5' region of this NBR2 is very similar to NBR1 gene, but the rest of the gene has nothing to do with NBR1. So end up this gene become a non-coding gene. So it does not appear to encode a protein. And then also there is a partial duplication of BRCA1 gene. So you end up with a BRCA1 pseudogene, which now resides right next to NBR1 genes. So in this locus, there are 4 genes. The BRCA1 gene and the NBR1 gene are protein coding gene, but the NBR2 and the BRCA1 pseudogene are non-coding genes. Now, this NBR2 gene, I should mention, is relatively long, is more than 1 kb. So by definition, it encodes a long non-coding RNA. So that's why we identify this NBR2 non-coding RNA from our analysis. And so we thought maybe because it resides right next to BRCA1 gene, we saw maybe it has some relevance to cancer biology. So we initially started to focus on this gene. So shown here is to validate that indeed glucose starvation can induce its expression in various cancer cell lines, but not in these two cell lines called A549 and HeLa cells. So if you don't know this, I should mention that both cell lines are LKB1 deficient cancer cell lines, meaning they don't express LKB1. So this caught our attention, suggesting that maybe the glucose starvation induced MBR2 is LKB1 dependent, because if you don't have LKB1, the cells do not induce the MBR2 under the glucose starvation condition. And this can be first validated by approach that if we put back LKB1 in those LKB1 deficient cells, now we can restore the glucose starvation induced MBR2 expression. Now we did some further analysis showing that if we inactivate MPK in the cells, again recording that MPK is a downstream target of LKB1, right? So if we inactivate MPK or using siRNA to knock down MPK in those MPK proficient cells, meaning they express MPK, now we can also repress the energy glucose starvation induced MBR2 expression. So together, this study suggested that LKB1 the glucose starvation can induce MBR2 at least partly through LKB1 and MPK pathways. So we still do not fully understand how MPK regulates MBR2 expression. So then we start to study the exact functional role of this non-coding RNA during the energy stress conditions, right? So we did a bunch of studies. We actually found it can feedback to regulate MPK. So shown here is a typical analysis we conducted to study energy stress response. So for example, you culture cells in the glucose-free medium, and you can potently induce MPK phosphorylation. So this is the one way to monitor MPK activation. As I mentioned, the LKB1 phosphorylate AMPK in response to energy stress. And correspondingly, you can also see the phosphorylation of another protein called acetyl-CoA carboxylase. So this is a key enzyme involved in lipid metabolism. So MPK can phosphorylate ACC, can inactivate ACC function, and you can see the ACC phosphorylation dramatically increase upon glucose starvation. Another protein called the raptor, which is a component of mTOR complex 1, it can also be phosphorylated by MPK, and you can see its phosphorylation is going up after under the glucose starvation condition. Now, correspondingly, as I mentioned, the MPK can repress mTOR complex 1 activation under the energy stress condition. So you see the S6 kinase and the 6 phosphorylation, which is a downstream target of mTOR complex 1. So those phosphorylation goes down upon glucose starvation. So this is a typical way we study this pathway. But what's striking is that if we knock down this non-coding RNA in cancer cell lines, and we see the glucose starvation induced MPK activation and mTOR complex 1 inactivation is significantly attenuated. So you see those induction is not very dramatic in the knockdown cell. Again, this downregulation is also attenuated in the knockdown cells. So suggesting somehow the MBR2 play a role in energy stress induced MPK activation and mTOR complex 1 inactivation. Now, in response to energy stress, MPK is activated. Then it functions to regulate autophagy, regulate cell survival, regulate cell proliferation, so a bunch of downstream cellular processes. So we tested further on those cellular processes and found they are consistent with the model that the MBR2 regulation of MPK. So basically the MBR2 can also regulate cell proliferation, apoptosis, and autophagy downstream of MPK on the energy stress. So I'm not going to show data here, but those data are all consistent with the idea that the MBR2 somehow can regulate MPK function during the energy stress. So these are so-called loss of function approach, meaning we deplete the gene in the cells, then we look at what happened, right? It's like if you study gene function in vivo, you can knock out the gene in the mice and look at what happened. Another approach complementary to loss of function approach is called the gain of function approach. So for example, you can also generate transgenic mice to study a function in vivo, right? So in cell line, we can also overexpress your targeted gene to see what happens. So here we basically overexpress different splicing variant of this non-coding RNA, and we found in all cases they function to promote MPK phosphorylation, but also downregulating the mTOR complex 1 activation. And correspondingly, we see a decreased proliferation if we overexpress those non-coding RNA in cancer cell lines, which is consistent with the MPK tumor suppressive function in cancers. So the next question is that how MBR2 regulates MPK. So we did a bunch of analysis. We did RNA pull down to identify its binding protein. So end up we identify actually the MPK is a binding protein of MBR2. So here I just show one key data. So the analysis here we did is called RNA pull down. So we use a biotin-labeled RNA to mix with the cell lysis. Then you use the streptovidin beads to pull down the biotin-labeled biotinated RNA and to look at what protein in those RNA precipitates. So in this analysis, we actually found there is a very striking glucose starvation-induced interaction between MBR2 with MPK. Now I should mention that there are three subunits of MPK complex. So if you pay attention, actually in the cartoon I showed earlier, the MPK actually there are three components called alpha, beta, gamma, in which the alpha is the catalytic subunit which contains the kinase domain. So in this assay, we found it can interact with all these subunits. But later, we further confirmed that this MBR2 directly interacts with MPK alpha, this catalytic subunit. So we further did analysis to characterize this binding. We show it is the N-terminal which contains the kinase domain interacting with the MBR2, but not the C-terminal. So that raises the question, maybe this non-encoding RNA by interacting with the kinase domain of MPK may actually modulate its kinase function. So we further tested this idea by we did a bunch of studies, but this just show one key experiment using the in vitro kinase assay. So basically use the in vitro purified kinase AMPK complex and to then examine its kinase activity towards some of its substrate peptide. So in this case, you can measure its kinase activity under the in vitro condition. So what we found is that if we add those in vitro synthesized MBR2 into this in vitro system, they can promote its MPK kinase activity to phosphorylate its substrate. So that's suggesting that MBR2 can somehow promote MPK kinase activity. So I'm going to skip a lot of data here because this is published in Nature Cell Biology last year. But I'm going to summarize the key finding here. That is, on the energy stress condition, as I mentioned earlier, MPK is activated. It can then induce. Our study shows that MPK can induce MBR2 expression. Then the MBR2 can form a feedback loop to potentiate MPK activation by interacting with MPK and can promoting its kinase activity. So that's our current model. So this overall form a feedback loop. So this non-encoding RNA can function to potentiate the MPK function during the long periods of the stress conditions. Now, does this have any relevance to human disease? And our data indeed suggests that. So for example, we found that in some human cancers, the MBR2 is downregulated. There is correlation between MBR2 expression with MPK activation in the human cancer patients. If we knock down MBR2 in tumor cells, that can promote the tumor development in the xenograft models. So that suggests that the MBR2 may also function as a tumor suppressor, similar to LKB1 as I mentioned earlier, to regulate the MPK and to monitor the energy stress response. So when there is mess up of this regulatory mechanism, that leads to tumor development. So that's the bottom line of this project, this study. So so in the remaining time, now there is some follow-up study from here. That is, here actually we touch on this drug called metformin and fenformin. I think for the students here, if you study diabetes, probably you know this. This is widely used drug to treat diabetes, and they actually mechanistically function as an inhibitor of mitochondrial respiratory chain complex 1. It can decrease blood glucose level, so it's one of the most widely used drug to treat diabetes. So why this has anything to do with our studies? Because many retrospective analysis plus now the clinical trials and also many functional studies support the idea that there is a beneficial effect of biguanide. So metformin and fenformin, they are structurally very similar as they together called the biguanides. So this type of drug in the cancer prevention and treatment. And mechanistically, at least one of the mechanisms is that because it functions as a mitochondrial respiratory chain complex inhibitor, so it can inhibit the OX4s, decrease ADP levels, increase MP level, so can activate MPK. Now I mentioned that MPK, once it's activated, can inhibit those anabolic processes such as protein synthesis. So it has been shown that metformin can promote MPK function, and thus inhibiting the cell growth and the cell cycle. Now, but on the other hand, in those cells with defective MPK, so if you don't have MPK in the cells, those cells, they have problem because normally the cell can adapt to the energy stress, right? So think about you don't have this checkpoint there. So those cells, they don't know whether there is energy stress, there is no energy there or not. So they continue to proliferate. So correspondingly, they are very sensitized to the energy stress induced cell death. Because you don't have energy, you continue to expand. Then what can you do? You have to die eventually, right? So it's like, you continue to expand, even you don't have money, then you have to eventually undergo bankrupt. So that's the same idea here. So that has proposed some ideas that maybe the metformin is actually particularly useful in those cancers which have defective MPK pathways, such as LKB1 deficient cancers. So in those cells, actually there has been studies show that the LKB1 deficient lung cancer cells are particularly sensitive to metformin or fenformin-induced cell death. So because we show that MBR2 regulate MPK, so we initially hypothesized that maybe MBR2 also regulate cancer cell sensitivity by guanylase, and we show that indeed the fenformin treatment here can induce the MBR2 expression. So this is similar to the glucose starvation-induced expression I just showed a couple of minutes ago. Again, we also show that the fenformin-induced cell death can be potentiated or can be enhanced in the MBR2 knockdown conditions. So that's consistent with the idea that this, because MBR2 can regulate MPK, so if you knock down MBR2, the MPK pathway is defective, right? So you would expect those cells has more problem with the fenformin treatment. But then here came with the problem. So we initially thought this is a very simple study, right? So we would confirm that MPK activation is downregulated in MBR2 deficient cells under the fenformin treatment condition. But what we found is there's no difference. So if you look at this data, which is very similar, very different from the glucose starvation data I showed a moment ago, that is, if we knock down MBR2, it does not have any effect on fenformin-induced MPK activation, ACC activation, or S6 phosphorylation downregulation. So you see there's no difference. So suggesting something else is going on here, right? So let me first show you why this is the case. This is because we eventually found that fenformin treatment does not affect MBR2 interaction with MPK. So I showed earlier that the glucose starvation can promote the interaction between MBR2 with MPK, but fenformin somehow does not affect this interaction. So that explains why we don't see any effect on the fenformin-induced MPK activation in those MBR2 knockdown cells. So then the question is, what's the mechanism to explain the effect of MBR2 on the fenformin regulation of the cancer cell death? Then end up we found that we did a bunch of study. I'm going to skip a lot of data, but to show that we eventually found that has something to do with this protein called GLUT1, which is a glucose transporter. So it transports uptake the glucose into the cells. So you can see here the fenformin treatment can significantly increase GLUT1 expression by either Western blot or QP cell. So that probably is because adaptive response of cells. So basically, fenformin treatment can deplete the ATP, right? Because it can inhibit OX4 process. But then the cells, they try to fight with this issue. They try to uptake more glucose by upregulating GLUT1 expression, so the cell can compensate with the fenformin-induced ATP depletion. But what we found is that this upregulation of GLUT1 upon fenformin treatment is almost abolished in those MBR2 knockdown cells. You can see very clearly that there is no increase whatsoever for the fenformin-induced MBR2 expression. Fenformin-induced GLUT1 expression. And also because GLUT1 play an important role in the glucose uptake, and we should also the MBR2 knockdown can also downregulate fenformin-induced glucose uptake. Now, functionally, we show that GLUT1 sensitization deficiency also sensitizes cancer cell to fenformin-induced cell death, which is consistent with a previous study from David Sabatini's group. We also show that conversely, if we restore GLUT1 in the MBR2 deficient cells, that can normalize the increased cell death upon fenformin treatment. So all these studies suggest a model that indeed the MBR2 regulates fenformin-induced GLUT1 expression and the glucose uptake, and GLUT1 play a role in the MBR2 regulation of fenformin-induced cell death. So here is the model. So in the first part of my study, I showed that glucose starvation can induce MBR2. MBR2 can interact with MPK, which can inhibit mTOR complex and to inhibit the tumor development. However, this is different under the fenformin treatment condition, even though both treatment induce energy stress. So we still don't understand why. But what we found is that baguanide or fenformin treatment can induce MBR2 expression. But MBR2, under those conditions, MBR2 does not regulate MPK, but instead upregulate GLUT1 expression. And this can promote glucose uptake and can help the cell to maintain survival under the baguanide treatment condition. Okay, so I'm going to take the last few minutes to focus on unpublished study from our recent project. That is to another non-coding we have studied on which is induced by transcription factor FOXO. So many of you probably know this because of, partly because the seminal study by Shaodong, in the early years to study FOXO. But FOXO basically is a transcription factor which play an important role at the crossroad between cancer and metabolism. Basically, this transcription factor play an important role on the two types of signaling pathway. First is the growth factor pathway. So in that case, the growth factor can activate the PI3 kinase AKT pathway. AKT can then phosphorylate FOXO and can sequester FOXO from the nucleus into the cytoplasm. So in this case, the growth factor inhibits FOXO activation. However, under the stress conditions, such as under energy stress conditions, the FOXO can also be phosphorylated by some stress-related kinase, such as MPK or JNK, on different sites. So once it's phosphorylated, the FOXO can then translocate from the cytoplasm into nucleus, can function as a transcription factor to upregulate many genes involved in the regulation of cell cycle arrest, apoptosis, or the detoxification of ROS. Now, I should mention that many FOXO targets as protein coding gene has been identified. Okay, but very few studies have shown that there is any non-encoding RNA which can be regulated by FOXOs. And I should mention that we previously already published a series of publications to study the FOXO important function of FOXO in mediating energy stress response or drug resistance, or in the context of kidney cancer. So here we continue to study its function in kidney cancer, but focus on its potential function to regulate any non-encoding RNAs. So approach we have done here is to use a cell line we established in previous study. We can turn on and turn off FOXOs by treating the cell with full HT. So this is similar with the tamoxifen treatment of CRE-ER2 to study the CRE activation to induce gene deletion. So in this case, we did RNA sequencing to compare the FOXO on and off conditions, and we did the RNA sequencing, identified many non-encoding RNAs which can be upregulated by FOXO. And one of the most striking non-encoding RNAs, again, we filtered with the TCG dataset. We identified a completely unknown non-encoding RNA which is significantly upregulated by FOXO, and we call it FOXO-induced non-encoding RNA number 1 or FLINK1. So just to show that we can, this non-encoding RNA can be very potently induced by FOXOs, and it is, we further show it's a FOXO target. It can be induced by glucose starvation in a FOXO-dependent manner. But what's really interesting is the function of this non-encoding RNA under the energy stress conditions. For example, we generated the knockdown cells of this non-encoding RNA in the kidney cancer cells. And if you culture those cells in different medium, you will see under the full medium condition, with glucose and glutamine and other nutrients, you don't have much difference. But under the glucose limiting condition, you see those cells they have much better conditions for the knockdown cells. And this can also be better appreciated from the image here. You can see if you culture cells in the glucose deprived medium, over time you see a substantial cells they're floating, right? So they died. But those knockdown cells, they are in much better shape. And we did further analysis to show that this is because those knockdown cells, they have less apoptosis in the glucose starvation conditions. So we further show that this has also relevance to cancer, because first, if we study the anchorage independent growth, which is in vitro analysis to kind of study the functional relevance to cancer biology, but you can see there is an increased soft agar formation of FLINK1 knockdown cells. But most the most striking data is from the xenograft model. You can see there is a very dramatic increase of tumor development of those FLINK1 knockdown cells in vivo. So suggesting that FLINK1 deficiency can promote kidney cancer development. Now, so the next question is, what's going on here, right? So we did a bunch of studies, and eventually we found is that actually this non-coding RNA can regulate glucose uptake and lactate production. So what's shown here is we found that the FLINK1 can regulate the many genes involved in glycolysis. So this is the pathway which can metabolize glucose to pyruvate, right? So once you generate pyruvate, it has many routes, downstream routes, but two fundamental one is the pyruvate can be metabolized to lactate, which can be secreted out of cell. The other is the pyruvate can be further metabolized to acetyl-CoA and then enter into TC cycles. So what we found is that many genes involved in glycolysis and also the lactate production or the secretion is dramatically upregulated in those knockdown cells. And this is associated with increased glucose uptake, increased lactate production, and also increased extracellular acidification rate. So this is indication of the Warburg effect. Before I go into further data, I want to show the relevance of this study to cancer. In this case, is kidney cancer. Because this, as I mentioned, we studied this non-coding RNA because it has some relevance from the computational analysis to link to kidney cancer. So here I show the data. Interestingly, this non-coding RNA is specifically expressed or mainly expressed in kidneys. So you can see it's very abundantly expressed in kidneys from various data sets. But interestingly, is significant downregulating in the kidney tumors. You can see in this case, this expression level is significant downregulating in the tumors, and we did a further analysis to show that this in the matched sample between the normal kidney and the kidney tumor samples. You can see in most of those kidney tumors, its expression is significantly downregulated compared with the matched normal kidney samples. Furthermore, you see there's an inverse correlation between this FLINK1 expression with those gene expression of the glycolysis genes. Finally, we show its low expression correlate. So you probably cannot see very clear here, but this blue line represents the low expression of this gene, which correlate with shorter survival. So again, this suggesting that this non-coding RNA may play a function as a tumor suppressor. So this is consistent with our mouse model to show we'll deplete this non-coding RNA, you see more tumor development. So now I'm going to talk about the just the last couple of minutes, the mechanism, how this non-coding RNA function to regulate glycolysis. So we found that this non-coding RNA can regulate the MYC level. So MYC, as you know, is a key transcription factor which regulates glycolysis and also play important role in tumor biology. So what we found is that if we knock down this non-coding RNA, we see a significant increase in MYC protein level under the glucose starvation. And I should mention that this knockdown does not affect the MYC mRNA level, does not affect its protein stability, but specifically affect its protein translation. I also show that the MYC largely mediates these biological effects afforded by FLINK1 deficiency on the energy stress conditions. For example, if we restore MYC in those, if we knock down MYC in those FLINK1 knockdown cell, we can largely rescue all those phenotypes. So then the question is how FLINK1 regulate MYC level, right? So here we have to use an unbiased way. Again, we used proton assay to try to identify the FLINK1 binding protein, and we found one of the binding protein called the AF1, which is particularly interesting because this is the AU-rich element binding protein, which combines to this called AU-rich elements containing protein, and actually MYC is one of such protein, these mRNAs. So it combines to the MYC mRNA with these elements and can promote MYC translation without affecting MYC mRNA levels. So this is consistent with the FLINK1 function as we just found, right? So we thought maybe because this FLINK1 combines to AF1 and regulates the MYC function. So this just to show that we indeed see there is an interaction between FLINK1 with AF1 protein. And again, this is very striking glucose starvation-induced interaction between the FLINK1 non-coding RNA with AF1 protein. So we still don't understand the detailed mechanism why would glucose starvation can induce such a striking interaction between non-coding and the protein. So we're still working on this. But this is very striking. We have repeated this many times, very consistent. Again, I skip some data here, but what we have found is that this FLINK1 interact with AF1 as shown here, can sequester AF1 away from binding to the c-MYC mRNA. So one assay to show this, if you, under the in vitro condition, you add more FLINK1 into the lysis, that can compete with the c-MYC UTR to binding AF1 protein. So if you see here, if FLINK1 can actually decrease the binding between c-MYC mRNA with AF1 protein. Conversely, if you knock down FLINK1, you can increase the binding between the AF1 protein with c-MYC mRNAs. So both analysis suggesting that the FLINK1 sequester AF1 from binding to c-MYC mRNA. So it's a lot of information, but this is a take-home message. I'm going to summarize the model here. That so what we have found that is under energy stress condition, the FOXO is activated. So FOXO nucleates in the nucleus and can regulate the FLINK1 expression. So once FLINK1 is upregulated, it functions as a kind of a molecular sponge to sequester AUF1, which is a mRNA binding protein, away from c-MYC mRNA. Then in the kidney tumors, the FLINK1 is downregulated. So in those cases, now the AF1 protein is available to binding to c-MYC, right? Because it does not have this molecular sponge to sequester it, to take it away. So now this AF1 protein is available to interacting with c-MYC to promote its translation. So more c-MYC protein will be generated, and more transcription factor, more c-MYC will function as transcription factor to regulate those glycolysis gene expression that can promote the Warburg effect, promote the tumor development. So that's our model here. So basically, this guy, FLINK1, functions as a molecular sponge to limit the c-MYC translation. Okay, so that can also inhibit kidney tumor development. So just the last minute, I want to just have a very broad view on how the potential function of non-coding RNA in cancer metabolism. Right? I mentioned that the non-coding RNA can function in the nucleus to regulate transcription by interacting with other epigenetic regulators. I also mentioned that it can function as a molecular sponge. So the FLINK1 is one such example to sequester its target protein away from binding to other biomolecules. But we envision that maybe some non-coding RNA can actually function as a sensor to sense the nutrient or energy levels. So in this case, it may can also one case is may can directly interact with those metabolic enzymes or protein kinase. For example, NBR2 is one such example. It can interact with NPK, can regulate its kinase activity. So we envision maybe there are other examples of such a non-coding RNA which can directly interact with those metabolic enzyme or kinase, can regulate their kinase or enzyme activity. And also we hypothesize maybe those non-coding RNA actually can function as a sensor to sense the availability of nutrient or the energy level in the cells. You think about the non-coding RNAs, they are RNAs, right? So they are very flexible in terms of the structure. So they may can adapt to some kind of structure to directly bind to those the nutrients and also corresponds to coordinate the nutrient availability to the downstream cellular processes. So this is a very intriguing hypothesis. I would say still hypothesis, but we are very fascinated by this hypothesis, and we're trying to test this hypothesis in our future studies. And finally, we also hypothesize maybe some of those non-coding RNA can localize in some of the intracellular organelles which play important role in metabolism, such as mitochondria, such as lysosome, and those intracellular organelle nucleus non-coding RNA may play also play important role in cancer metabolism, which will be another interesting hypothesis to test in future studies. So with that, I'm going to end my talk to thank my lab members, particularly Zhendong Xiao and Xiaowen Liu, who have focused on their study on the non-coding RNA function in cancer metabolism, and our collaborators, and also various funding support in our studies. Finally, I want to thank you all for your attention, and I would like to take any question if you have. Thank you.
Questions?
So I
Yeah.
have a more general question about the non-coding RNAs.
Yes.
So how are they normally, are they constitutively transcribed, or are they
So I think you can think about this as a non-coding RNA just as the protein coding gene. So for protein coding gene, we have learned a lot, right? Its transcription can be regulated, very precisely regulated, right? So it can be very dynamic. Its transcription can be very dynamically regulated. So it can be up-regulated or down-regulated upon different type of stress or stimulus. So in this case, the non-coding RNA similarly can also regulate by different types of signaling pathways.
So my question is related to the NLB2.
Right.
Is in proximity to the BRCA.
Right, very close,
So
yes.
would that be under the same transcriptional regulation as the BRCA gene, or is that totally different?
Right, very good question. So we found that in this case is the glucose starvation seems does not regulate the BRCA1 gene expression under the same conditions. So suggesting that those regulatory elements may be different in terms of how their transcription are controlled by glucose starvation. Yeah. So that can be probably we need to do further analysis to tease out exactly the elements, the DNA elements which control the transcription of MBR2 under the glucose starvation condition. Yeah. But looks like they're different. Yeah.
So following that, so it looks like so it's pure speculation.
Right.
So you expect that for every oncogene there will be a long coding RNA to act as a tumor suppressor gene for those.
Could be, yeah, I think, yeah. So, usually we call it yin and yang, right? So, I think more broadly is we probably, so if we only talk about the tumor biology, when there is oncogene, there always, in most cases, I would say there's also a corresponding tumor suppressor gene. So, usually we call it yin and yang. So, for example, that's how, for example, in this case, the TSC was identified. So, TSC basically is a yin. But the yang could be PI3 kinase or mTOR. So people first identified PI3 kinase and mTOR. And they don't know, for example, in the early years, they don't know the negative regulator of mTOR. So that's how they identified the TSC, which actually functions as a yin of mTOR. But there could be also, so if you extend these concepts, so it is possible that if there is oncogene protein, then there's corresponding negative regulator of this protein, but those negative regulator could be non-encoding RNA. So they can function as a yin, but function as a non-encoding RNA to regulate the protein the protein oncogene. Yeah. Yes.
I'm interested that you used fenformin rather than metformin, given that fenformin isn't used at all clinically.
Yeah, great question. Yeah, so that's a very, very good questions. So as you probably know that what was widely used in the patients is metformin, right? So fenformin has been, I think, taken off from the market because it has some side effects, right? Right. Yes, very good question. So in our study, and also has been shown in many other studies, is that the fenformin is much stronger the mitochondrial respiratory chain complex inhibitors. So first, it has in general has more potent effect in terms of induced cell death. But more importantly is the fenformin. The cells need a transporter called OCT1 to uptake metformin. But the fenformin is more hydrophilic, so it does not need those transport. It can directly cross the membrane. So that's why in many cell lines, if you treat with metformin, you might not see effect because they're largely dependent on the expression level of this transporter.
But doesn't metformin get taken up a lot more by OCT2, and so then there's different tissue distribution of OCT2s? I'm just wondering how translatable your
Right. So great question. So actually now, so this you mentioned this, right? Fenformin has some side effects in patients, but from the cancer treatment perspective, the side effect actually is quite minor. I think there is, I forgot the exact rate, but so the patient who have this kind of side effect, the rate is the percentage is very low. So if we compare, there has been an argument that if we compare this side effect with the side effect of those traditional therapeutic treatment in cancer patients, so this actually is a much low side effects, compared with other more classic drugs.
So there is. This could
So there's interesting now to use the fenfluramine treatment in cancer patients. So actually there's now is phase 1 clinical trial to test the fenfluramine in cancer patients. So bottom line is, yes, there is side effect, but now also compared with other treatments, this side effect is considered to be moderate, and there is a growing interest to use fenfluramine in the treatment of cancer patients.
So have you done your same experiments with metformin to see if that
With
reduces it?
Right. So it largely depends on the cell line. For example, in the cell line we used here, we don't see the obvious, for example, the cell death effect if we use metformin. So again, we think this might relate to the expression of the fenfluramine, the uptake, the transport to uptake fenfluramine in the cell line we have been studying. And this has been also shown in many other studies. Yeah. So that's why many studies actually they also use fenfluramine. Yeah. Great question. Yes, A. Yeah.
That's a great talk. So related to MPK and the NBR1 meta-interaction, you said
NBR2, right.
actually intact with the kinase domain of MPKR.
Right.
So because it's a serotonin kinase, and this kind of kinase are really similar to other kinase.
Uh-huh.
So the question is that do you know if this NBR2 can also target other kinase domains?
Yeah, that's a great question. So we haven't tested this yet. Yeah, I think also this relates to a more fundamental question, on the biochemical or structural, biology point of view. What's the exact, what's going on there, right? How a non-coding RNA can regulate the kinase function. So we haven't been able to test other kinase. So, but that's going to be, I think, a very good question to test in our future studies. Yeah.
So you're planning to look at the
Right. Yeah, it's challenging. Yeah. But we also, yes, we are indeed interested in pursuing this. Yeah.
So probably you mentioned this is NBR2 mutated or deleted.
It's mainly downregulated in kidney cancer and breast cancer. Yeah. And actually, it's low expression also correlated with poor clinical outcome in those cancers. Yeah. Yes.
Just very quickly, you showed the very elegant data about the non-coding RNA that have very different effect under high glucose, low glucose.
Right.
Which may make me thinking about if anybody have studied the non-coding RNA in the setting for type 1, type 2 diabetes, and whether,
Great question. Yeah, we haven't. I think we haven't done this. Yeah, I mean, we hope any experts in this, studying diabetes probably can test this further. Yeah, and I should mention that one issue is, I think also one challenge to study non-coding RNA is relatively, so those non-coding RNA are less conserved in other organisms. So meaning if you identify a non-coding RNA from human cells, it's very likely they are not conserved even in mouse. Or if you identify non-coding RNA in C. elegans or mouse, they may not be conserved in humans. So they have a relatively poor conservation cross species. So in this case, for example, for NBR2, we cannot rule out the possibility they may be conserved, but at least right now we don't see obvious orthogonal gene in mouse. So that makes it more challenging to study, for example, its function in mouse. If we study, for example, diabetes, oftentimes we have to generate transgenic mice or knockout mice. But then in this case, because we don't see an obvious orthogonal gene of NBR2 in mouse, so that makes currently difficult to study its function in mouse model.
Probably in the Alice culture or.
Yes, definitely it's expressed in human. Yes. Yeah.
Okay.
Yes.
Well, let's talk one question. So, in cancer treatment, generally, the glycolysis inhibitor is trying to be used to block the warp effect,
Right.
to inhibit cancer growth.
Right.
Like metformin actually is the opposite, which can promote glucose uptake, promote glycolysis, but still inhibit the tumor development.
I think it is because so its primary function is to inhibit the mitochondrial, right, respiratory chain complex, right, so inhibit the ATP production. But this glucose uptake, I think, is a compensation, is a compensatory effect. So that actually, you could argue maybe you combine the metformin or fenfluramine with a glycolysis inhibitor, you may have an even better clinical outcome. Yeah. So that's the general issues with many cancer drugs, because it can, when you treat the cells with drug, it can also stimulate some kind of compensatory effect, by activating, for example, PI3 kinase FOXO. So if you inhibit the AKT or PI3 kinase, it can reactivate actually some of upstream receptor tyrosine kinase. So that can bring some issues. Yeah.
Okay.
a student, you are invited to lunch. Even if you're not enrolled in the class, I'll just remind you of that. That will be held directly following this over in our building. So please make sure you do that, and please join me in thanking Dr. John again.
Thank you.
Do you have any pin drive?
I think is I upload one here. Maybe I should delete it. Right.
This one?
We'll just delete it.
Oh, just delete it?
Just delete it.
That's
Hey.
a really great talk.
Yeah, thanks. So I will meet with you
Yeah,
tomorrow. Right?
yeah,
Yeah, good.
Talk
How is
to you
everything
later.
going?
So far,
Yeah.
so good.
Okay. So we'll talk more. Okay, later. Yeah. So just delete it. Okay.
Go ahead.